Hello, nurses. We're going to use this time to cover preliminary material, in this case, cells, tissues, organs, and so on. So the cells of our bodies are eukaryotic cells. Bacteria are prokaryotic cells. Our cells are eukaryotic cells. Prokaryotic cells do not have any internal compartments. Eukaryotic cells have an abundance of membrane-bound internal compartments. The functions of the cell tend to be localized to those membrane-bound compartments. So each of these compartments that you see here has a specific function. We'll go through each of the functions and what they do and their importance for the life of the cell. So the first of these is the lysosome. The lysosome contains a group of enzymes called acid hydrolases. Acid hydrolases. The membrane of the lysosome has a hydrogen ion pump that pumps hydrogen ions into the interior of the lysosome. That acidifies the interior of the lysosome. As the lysosomal interior becomes acidified, those acid hydrolases become activated. These acid hydrolases degrade things like proteins, lipids, complex sugars, in some cases, nucleic acids. They degrade lots of the things that the cell might ingest, but the cell can't digest. The lysosome does the digestion for the cell. So it takes macromolecules, converts them into smaller molecular precursors. Those are then exported to the cytoplasm, and then the cell will use them or export them as needed. Anything the lysosome cannot degrade will usually be exported by a process called exocytosis, and that garbage, as you will, gets just taken out. Lysosomes are derived from the Golgi apparatus. If we take a look at our slide, and if we use our pencil here, so here's the Golgi apparatus right there. The Golgi apparatus receives materials from the endoplasmic reticulum right here. The enzymes that will make the interior of the Golgi and the membrane proteins, they're all exported from the endoplasmic reticulum to the Golgi, and then they will get together in a bud. The lysosome will bud, and then the lysosome goes through stages of maturation. Mutations in structural genes for these acid hydrolases will prevent the cell from being able to degrade certain macromolecules. Those macromolecules will build up inside the cell and kill the cell. Those group of genetic diseases are called lysosomal storage diseases. These include things like Tay-Sachs disease, Sandoff disease, Hurler's disease, and the like. Peroxisomes are a more independent membrane-bound organelle. So these membrane-bound structures are called organelles. Organelle means little organ. Peroxisomes are used to degrade long-chain fatty acids and to detoxify complex organic molecules. So liver cells will have lots of peroxisomes. Anything that does a lot of fat metabolism will have a lot of peroxisomes. Peroxisomes will have their own machinery for importing and exporting molecules. These are bound by a lipid bilayer. They will generate lots of reactive oxygen species, things like hydrogen peroxide, superoxide radicals. Consequently, peroxisomes have lots of catalase that takes hydrogen peroxide, converts it to water. Another enzyme called superoxide dismutase converts superoxide radicals to hydrogen peroxide, and then the hydrogen peroxide gets converted to water by catalase. So lots of this detoxification occurs inside peroxisomes. Problems with superoxide dismutase have been linked to sporadic types of amyotrophic lateral sclerosis, or ALS. This is a nervous system condition that causes defects in motor function and then sensory function as well. Many nervous system functions will affect just sensory or motor functions. ALS is unique in that it affects both. Ribosomes are not membrane bound. These are the machines of protein synthesis inside cells. If we look at a ribosome up close and personally, we can see that there is a small ribosomal subunit. This small ribosomal subunit is called a 40S ribosomal subunit, 40S. This consists of RNA and protein. The RNA is called ribosomal RNA, that is abbreviated rRNA. It's very hard to write with a mouse. So this plus these ribosomal proteins, they assemble together to make the small ribosomal subunit. The large ribosomal subunit is a 60S ribosomal subunit. That's the large ribosomal subunit. This contains ribosomal proteins and then several significant ribosomal RNAs. In particular, there is the so-called 28S ribosomal RNA, which is found in the large ribosomal subunit. 8sRRNA. When these two ribosomal subunits assemble together, they form something called an 80S ribosome. Now, you might be saying, well, look, 40S plus 60S, that should be 100S. You're right. This is an 80S structure because this number here, S, that is a Svedberg. A Svedberg is a unit of descent in a centrifugal field, and that depends on size and shape. So when these two subunits come together, they make something that doesn't descend quite as fast because it's not as streamlined. The ribosome is responsible for protein synthesis. Protein synthesis in cells is called translation. Translation requires messenger RNA. So here's a messenger RNA. Messenger RNAs are made in the nucleus. And these messenger RNAs go to the ribosome. The ribosome reads them. It reads them from front to back. The front of the messenger RNA is called the 5' end. That's the front 5' end. The 3' tail is over here. And the protein, therefore, gets made, it gets encoded on the messenger RNA, and it gets made from front to tail like that. The front of the protein is called the amino terminus. That's the front. So that's the first part of the protein that gets made. Proteins are made from amino acids. Those amino acids are brought to the ribosome by tRNAs, transfer RNAs. Transfer RNAs are also made in the nucleus, processed in the nucleus, and then brought to the cytoplasm. The appropriate amino acid is linked to the appropriate tRNA by a special group of enzymes. And those special groups of enzymes are called amino acyl tRNA synthetases. Very interesting group of enzymes. Been a lot of work done on those enzymes. Aminoacyl. Excuse me. tRNA synthetases. Maybe one of these days I'll figure out how to use my pen on my iPad and do this on my iPad so I can use my pen. So these enzymes, there's one specific enzyme for each amino acid, and it's cognate tRNA. They read this sequence here on the tRNA. This sequence here is called the anticodon sequence. And then they read the acceptor loop, and they attach the amino acid to the appropriate tRNA. It's called the anticodon. And then the anticodon, as you see, has a sequence that's complementary to a 3-base sequence on the tRNA, and that sequence is called the codon. And that's how the ribosome knows it's putting the right amino acid at the right part of the protein. And then the 28S ribosomal RNA is actually an enzyme. It's a ribozyme enzyme, and it forms these peptide bonds between the amino acid. And each amino acid is then added sequentially, and that's a process of translation. You need ribosomes, messenger RNAs, charged tRNAs, and then lots of other proteins that will help with this, that help initiate, maintain, and terminate this process. That's a process of translation. Proteins that will begin their lives by being made at the endoplasmic reticulum are proteins that are either secreted, are embedded in membranes, or are going to be lysosomal proteins or something like that. These ribosomes that are making these membrane-bound or secreted or lysosomal specific proteins, they will attach to the endoplasmic reticulum. They'll either embed the protein in the membrane, or they will secrete it into this cisternal space of the endoplasmic reticulum. And the endoplasmic reticulum will then assemble these proteins into a vesicle. That vesicle will bud from the endoplasmic reticulum and go to the Golgi apparatus. So this ribosomal decorated part of the endoplasmic reticulum, this is called rough endoplasmic reticulum. The other thing the endoplasmic reticulum does is that it makes lipids and it detoxifies certain types of xenobiotics, that is, foreign molecules. The enzymes for xenobiotic processing, lots of drugs are xenobiotics. Those enzymes are localized in the smooth endoplasmic reticulum. So liver cells, for example, have lots of smooth endoplasmic reticulum. They contain enzymes for lipid synthesis, cholesterol biosynthesis, and cholesterol handling, things like that. They also have enzymes called cytochrome P450s. And those enzymes, they do a lot of the handling of xenobiotics. And those are found predominantly in the smooth ER. Biotics. Yeah, I know it looks like my drawing is that of a 6th grader, or 1st grader, but it's just hard to draw with a mouse. As I said, most of the vesicles from the endoplasmic reticulum will go to the Golgi apparatus. They will come in here on the cis side, In the Golgi apparatus, these proteins will be modified further, and then vesicles will bud usually from the transphase, and they will either go to the membrane for secretion, or they will bud and become lysosomes. Some of these vesicles will be automatically secreted. Others will be held until the cell gets a signal for them to be secreted. And then some of the proteins that come here are meant to stay in the ER, so they actually come in and then they go back to the ER. So the ER plus the Golgi apparatus is the FedEx of the cell, pretty much. One thing I failed to mention with the ER is always contiguous with the outer nuclear membrane. And we'll say more about that here in a bit. Mitochondria have two sets of membranes, an inner mitochondrial membrane here, and an outer mitochondrial membrane here. And then where I put the X here, that's the intermembrane space. Within the inner mitochondrial membrane is the matrix. The matrix, obviously named after the movie, right? Yeah, not really. Now inside the matrix, the mitochondrion has its own ribosomes and its own genome. And both these genomes and these ribosomes are very bacterial looking. Bacterial looking. The genes are organized like they're bacterial genes. This genome, mind you, is very small. There's multiple copies of it also in the mitochondrial matrix. The inner mitochondrial membrane has a whole host of proteins that compose the so-called electron transport chain, which I'm just going to abbreviate ETS or ETC, electron transport chain. The mitochondrion is the site where cells make the lion's share of ATP. So this is your power plant of the cell. That's not all mitochondria do, but they make the energy of the cell. If mitochondria go south, the cell is in big trouble. If mitochondria get ruptured, that's usually a signal to the cell for the cell to undergo programmed cell death. So the integrity of the mitochondrion is very important for cells. Cells are delimited by a plasma membrane. The plasma membrane, as you can see, is a phospholipid bilayer. These phospholipids are based on the molecule glycerol. So glycerol has 3 carbons like this. And then it has 3 hydroxyl groups like this. And then lots of hydrogens coming off like this and blah, 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 what have you, because there have to be 4 bonds to a carbon, as you might remember from GLB. Esterified to these 2 alcohol groups here are fatty acids. So fatty acids, you might remember from GLB, they have a C double bond O. So there's a carboxylic acid group here, and then you have some sort of aliphatic chain here, which I'm just going to say CH2, and there's lots of these. So this one here with 3 carbons, that's propionic acid here. These are usually 16 carbons, 14 carbons, 18 carbons, 22 carbons, something like that. They may or may not have double bonds in them. So if you say take water away from this, you're going to end up with an ester bond. And you have two fatty acids esterified to these two bonds. To this bond, to this alcohol, you have a phosphate that's attached. And the beauty of phosphates is that they're charged. So this part of the molecule is charged. This part of the molecule here is very water-hating. So these two straight lines here, those are fatty acids. That doesn't like the water. Let's pick a different color here. This piece here, that's a piece with the phosphate, the glycerol bridge with the phosphate. Usually there's something else attached to the phosphate called a head group. That head group will vary depending on your phospholipid. Could be choline, could be ethanolamine, could be the amino acid serine, could be the sugar inositol. Those are the types of things that the head group might be. The two hydrophobic bits face each other, and then proteins are inserted into this phospholipid bilayer like icebergs floating in a phospholipid bilayer C. Proteins usually, if they face the outside of the cell, are usually decorated with sugars, and therefore they're called glycoproteins. Glycoproteins are simply proteins that have attached sugar groups. And many of the proteins embedded in the plasma membrane, the bits of the protein that face to the outside have these sugar attachments, so they are glycoproteins. In some cases, you have lipids that have sugars attached to them, almost always facing the outside, almost never facing the inside. These are called glycolipids. Good example of a glycolipid we have, particularly in red blood cells, are the cells responsible, the glycolipids responsible for the ABO blood groups. Those particular different types of ABO blood groups are due to distinct glycolipids that are found on the surfaces of red blood cells. And then you also have embedded in the plasma membrane and other membranes in your cells is this thing called cholesterol. Cholesterol has a steroid nucleus. Cholesterol regulates the fluidity and stiffness of the membrane. Cholesterol is also an important precursor for the synthesis of vitamin D and for all steroid hormones in your body. So hormones are made by the adrenal cortex. They're all steroid hormones. They're made from cholesterol. Your gonads make steroid hormones, estrogen, progesterone, testosterone, other types of androgens, other types of molecules like that. They are made from cholesterol precursors as well. Proteins inserted in the membrane are called integral membrane proteins. If a protein just hangs out near the membrane but is not embedded in it, that's called a peripheral membrane protein. Both kinds are important. Cells stick together. They stick together by means of these things called cell junctions. Cell junctions. When cells are stuck together, they form this structure called an epithelium. There are different types of epithelia. You investigated that in AMP last semester. There are squamous epithelia where the cells are flat like pancakes. There are columnar epithelia where the cells are nice and tall. There are cuboidal epithelia where the cells are as tall as they are wide. Those cells are pressed together and stuck together by junctions. These junctions are found at the apical sites of the cells. So they're often called apical junctions. There are two of them. First, there are tight junctions. Tight junctions. This year in A&P, you're going to look at the kidney. And the kidney has, in parts of the nephron, there are very important tight junctions that seal those cells together very tightly. The tight junctions are the apical-most junction. You have proteins called ZO proteins. That stands for zonula occludens proteins, or ZO1, ZO2, what have you. The ZO proteins are similar to proteins that were discovered in the lab I worked in for my PhD at UC Irvine. The dislarge proteins in the fruit fly Drosophila, they are the orthologous proteins of the ZO proteins. The ZO proteins seal these tight junctions together. Below the tight junctions, you have these things called adherens junctions. And adherens junctions are formed by these proteins called cadherins. There's lots of different types of cadherins. So E-cadherins tend to be found in epithelia. That's why they're called E-cadherins. Neurons, where they tend to stick together, they have something called N-cadherin. N-cadherin. And there are lots of different types of cadherins. Big thing you need to know about cadherins is that cadherins rely on calcium in order to be able to properly adhere cells together. Cadherins stick together with other cadherins. And these junctions that they form essentially work with the tight junctions. And one of the first things that happens often in cells that are going to become cancer cells is that they stop expressing E-cadherin. They stop making adherens junctions. And so the cells fall apart and they don't form proper epithelia anymore. And these connections are very important for the controls on cell proliferation. Attached to the adherens junctions are elements of the actin cytoskeletal network. We'll have more to say about that in a bit. These actin networks form belts across epithelial cells. These belts are under tension. That tension is extremely important for regulating what goes on in the cell. Now, at basolateral membranes, so if this is apical membranes, if these are the so-called ipsilateral membranes, you have ipsibasal membranes, basal membranes, ipsi-apical membranes, and then the apical membranes up here. So these would be your adherens junctions here. These would be your tight junctions up here. Down here, cells adhere together and then they adhere to basement membranes. These structures here are called desmosomes. Desmosomes. Desmosomes are really important. Desmosomes attach cells to substrata. So wherever you have epithelial cells that attach to substrata, those are attached by desmosomes. Desmosomes, as you see, have particular types of linker proteins together. You then have other types of cytoskeletal elements that attach to desmosomes. Those are called intermediate filaments. Also, usually at these basolateral membranes, you have these junctions that connect cells together called gap junctions. And gap junctions, as you can see here, allow the passage of small molecules between cells. These are extremely important in highly conductive cells. For example, cardiomyocytes that compose the myocardium. These gap junctions allow cardiomyocytes to experience ionic changes that instantaneously flood from one cell to the next. Therefore, the myocardium will work as a unit. Now we come to cytoskeleton. So the cytoskeleton is the rebar. So if you've ever worked with concrete, concrete has to be reinforced with rebar. Otherwise, if you put the concrete under shear forces, it will crumble. This is the rebar of the cell, the cytoskeleton. There are three types of cytoskeleton. There are actin, microfilaments, tubulin microtubules, and intermediate filaments. So here is an actin microfilament. You can see it is composed of actin monomers. The actin monomers assemble into a two-strand helical structure. These sometimes can be long-lived, and they will support apical structures like microvilli at apical surfaces that give cells more absorptive surface area. Their formation can also be very transient when cells are moving. And if you have motile cells, like white blood cells or something like that, this actin microfilament network is constantly being remade. And this allows the cells to be able to move because the microfilaments will form at the leading edge of the cell, pushing the cytoplasm ahead and then retracting from the back, thereby picking up the cytoplasm from the back and moving it in the direction the cell is going. Very important part of the life of the cell, actin microfilaments. This is a tubulin microtubule. It's composed of tubulin dimers. The tubulin dimers are alpha and beta tubulin. These dimers assemble together to form this hollow tube. Tubulin microtubules form large filaments inside the cell, and they provide a network by which organelles can attach and move. There are proteins that bind to tubulin microtubules and allow things to move one way or the other. The end of the microtubule that's growing is called the plus end. The end that is either not growing or that is diminishing is called the minus end. There are minus end microtubule motors. Kinesin is an important one. Excuse me. Dynein is a very important one. And then other microtubule motors, for example, kinesin. That will bind and carry things to the plus end of the microtubule. Microtubules are extremely important during cell division. They will bind to chromosomes and pull the chromosomes apart. And so that leads us to a discussion of the nucleus. The nucleus houses the chromosomes. Chromosomes are made of DNA. Chromosomes in our cells are linear DNA molecules. In mitochondria and in bacteria, those DNA molecules are circular. In our cells, our chromosomes are linear DNA molecules. They are assembled into a complex 3D structure called chromatin. They are assembled with the help of proteins called histones. That compacts the DNA into a very tight structure that can fit inside the nucleus. Gene expression occurs inside the nucleus, and Processing of RNAs occurs inside the nucleus. The site where ribosomal RNAs are synthesized inside the nucleus is called the nucleolus. So that's a site of ribosomal RNA synthesis. Nucleus is delimited by a dual membrane nuclear membrane, which I'm just going to call NM. And this nuclear membrane is decorated with these nuclear pore structures. These nuclear pore structures are very interesting. There's a paper that just came out in Nature Cell Biology that shows that those nuclear pore structures actually determine in zebrafish embryos when the embryo starts its own gene expression. Very, very interesting work. Here's the structure of DNA. DNA is a polynucleotide. So a nucleotide has 3 parts. It has a phosphate. It has a ribose sugar, and it has a nitrogenous base. Put those 3 together, you have a nucleotide. If you link them together, these nucleotides together, you have a polynucleotide. DNA is a double-stranded polynucleotide. One of these strands goes from this direction, from back to front. The other one goes from front to back like this. We call that arrangement antiparallel. Antiparallel. The bases are complementary to each other. That means whatever base you have here, you have a corresponding base here. If you have a cytosine here, you'll have a guanine here. If you have an adenine here, you have a thymine here. Thymine here, adenine here, so, guanine over here, cytosine over there. So you have A, T base pairs and C, G base pairs. These are linked together by hydrogen bonds. There are 2 hydrogen bonds holding each adenine thymine base pairing together, 3 hydrogen bonds holding each cytosine guanine pairing together. Gene expression is mediated by this enzyme RNA polymerase. There are 3 different types of RNA polymerases in eukaryotic cells. There is RNA polymerase 1, RNA polymerase 2, and RNA polymerase, you guessed it, 3. And these have such boring names because they were named by biochemists. Only a biochemist could come up with a name quite so boring. RNA polymerase 1 makes all the ribosomal RNA, well, almost all the ribosomal RNAs. So it is very active at the nucleolus. RNA polymerase 2 makes your messenger RNAs. RNA polymerase 3 makes tRNAs. Makes one of the ribosomal RNAs, makes the 5S, the smallest of the ribosomal RNAs. And then it makes a variety of small RNAs. Some of these are not terribly well understood, but RNA polymerase 3 makes them. Now, to do this, it has to bind to the DNA. It binds to the DNA at a site called the promoter. Doesn't bind just any old place, binds to a specific sequence called a promoter. The promoter has a whole bunch of proteins bound to it, and those proteins will tell RNA polymerase, hey pal, bind here and start here. These proteins are called transcription factors. It binds, it locally unwinds the DNA, as you can see here. It then starts making its messenger RNA, and then it proceeds essentially to make the RNA, unwinding as it goes this in the direction in which it's going, rewinding in the direction in which it's proceeding, and putting in the proper base. There is a very powerful proofreading function in this enzyme. If it puts the wrong base in, as it makes this RNA, it keeps getting checked. If it puts the wrong base in, it stops, backs it up, chips that errant base out, restarts the process again. Very accurate enzyme. Then there are sequences in the DNA that tell it to stop. When RNA polymerase gets to those sequences, it stops. It rewinds the DNA, and then the RNA, and this is a nascent messenger RNA, will then be completely processed. Mind you, much of the processing occurs while the messenger RNA is being transcribed, but that processing has to occur, and then it gets bound by groups of proteins and then goes to the cytoplasm for translation. That's the case if it's RNA polymerase II. If it's a ribosomal RNA, it gets highly processed after it's made. Then it assembles with ribosomal proteins. Then it gets shipped to the cytoplasm. tRNAs also get processed, and then after they get processed, they bind to transport proteins and then go to the cytoplasm. And there are a few RNAs that are made that stay in the nucleus, in particular the small, your microRNAs. They stay in the nucleus in some cases. In some cases they don't, in some cases they do. MicroRNAs are made by Pol II. Nuclear pores are embedded in the nuclear membrane. The nuclear pore dictates what enters and what exits the nucleus. The nuclear pores also are a binding site for a crucial set of proteins that give the nucleus its structure. And these proteins are shown here. These are called nuclear lamins. Nuclear lamins. These nuclear lamins form a network beneath the membrane. They interact, as you can see, with chromatin and several other proteins that are very important for the job the nucleus does. These consist of nuclear lamin A and B and C. And during cell division, the nucleus dissolves because these get phosphorylated. And then after cell division, these are dephosphorylated and the nuclear membrane reforms. Several different proteins from the cytosol interact with proteins that are embedded in the nuclear membrane. And in many cases, shear forces that occur on cells get transmitted to the nucleus, and sometimes those shear forces will actually influence gene expression. This is very clear in the case of tenocytes, which are found in tendons. Tenocytes have to be under tension. And if they're not under tension, they throw a fit. That tension influences their gene expression. And if they're not under tension, it might mean that the tendon's damaged and these cells need to start dividing. And this essentially shows you that these lamins provide this structure that allows the nucleus to properly communicate with the cytosol. And this is just a little network that shows you have your lamins here. And look at all the proteins that interact with these lamins via these networks. So lamins don't just stay in the nucleus. They're very important for nucleus cytosol interactions. I don't expect you to know any of these. I just want you to know that lamins are not by themselves. And then when it comes to genetic diseases, several different genetic diseases are known to involve either lamins or lamin protein, cytosolic protein interactions. And this shows you essentially some of the interactions of some of those proteins and the genetic diseases they might cause. So here's a nuclear pore complex. These are very complicated structures. It's very simplified here, but that's okay. There's kind of a spoke-like structure on the inside here. There are fibrils that jut this direction, and it seems pretty clear that what's ever going to get into the nucleus has to be able to bind these. These consist of proteins that are called FG proteins, because they have these two amino acids, phenylalanine and glycine, that repeat over and over and over and over and over again. Proteins that mediate import into the nucleus are called importins. As you might guess, importins are important. And importins bind their cargo. Here's their cargo, whatever it is. And they're able to bind to the fibrils. And that allows them essentially to get the cargo into the nucleus. Then there are proteins inside the nucleus called exportins. And exportins will bind their cargo and negotiate their way like this and get whatever they're trying to get into the nucleus out into the cytosol. Cells go through the cell cycle. Our cells go through a proper cell cycle. The cell cycle consists of multiple parts. M phase is the actual mitotic part of the cell cycle. From this part, M phase, through here, all of this here is interphase. That's interphase. Mitosis essentially just consists of this phase here. So if we were to break the cell cycle into a 24-hour day, this would be 1 hour, and the rest of the time, interphase would be 23 hours. So the cell spends most of its time in interphase. Interphase consists of 3 phases: the G1 phase, the S phase, and the G2 phase. S phase stands for synthesis. Synthesis of what? DNA. This is the only time during the eukaryotic cell cycle when the cells will replicate their chromosomes. G1 and G2 are growth phases. During G2, the cell is determining if the DNA was accurately replicated. If it wasn't, it will repair it. And if it's not accurately replicated, the cell will not proceed into M phase. During G1, the cell is determining if it has proper resources to synthesize DNA, because making DNA takes a lot of resources. If not, the cell will go into a quiescent phase called G sub 0. During G sub 0, the cell takes a nap. As you can see here, progression through the cell cycle is dictated by these proteins called cyclins. And there are different parts of the cell cycle that are specific for different aspects. Different cyclins are associated with different parts of the cell cycle. Cyclin B is associated with M phase. Cyclin D is associated with early to mid G1. Cyclin E is associated with late G1 and S phase. Cyclin A is associated with emergence, late S phase and emergence from S phase, and early G2. And then cyclin B is associated with progression into M phase, and then degradation of cyclin B is associated with emergence from M phase into G1. Incidentally, disruption of cyclins is associated with certain cancers. Biological membranes will let very small polar molecules through, like water. They'll also let small hydrophobic molecules through, like water, and small hydrophilic molecules through, like steroid hormones. They will not let charged molecules through, so ions can't get through. So cells, if they find themselves in a situation where there are more solutes outside the cell than there are inside the cell will give up water. And that pressure on the cell pushing the water out, we call that oncotic pressure. Now, hydrostatic pressure or osmotic pressure is typically measured as the force required to resist the movement of that water from the cell with fewer solutes to the solution that has more solutes. Also, if the cell finds itself in a situation where there are very few solutes outside the cell, then water will rush into the cell. And that, once again, that in this situation we have low oncotic pressure inside the cell, so the cell can't resist the influx of water. And this is related to these things called hypertonicity, hypotonicity. So hypotonic solutions like distilled water will cause cells to swell up. Hypertonic solutions, like a 2 molar sucrose solution, will tend to cause a cell to give up water and tend to cause a cell to shrink. Isotonic solutions, which you'll use in your IVs, like D5W, that's 5% glucose, like NS, normal saline, which has about 0.9% salt, or Ringer's lactate or something like that. All of these are isotonic solutions to where the net gain and loss of water in cells that receive these types of solutions is 0. Those are isotonic solutions. If cells have to give up energy to bring something into the cell, that's called active transport. If the cell does not have to give up energy, that's called passive diffusion. Or in some cases, passive transport, because cells can take up some small molecules by something that does require a carrier. That's called facilitated diffusion. But shown here, if cells have to output energy to get it, that's active transport. There's 2 types of active transport. There's primary active transport, where we have the direct hydrolysis of ATP. And the direct hydrolysis of ATP will pump molecules against their concentration gradients. And virtually all your ion pumps are primary active transporters, like your sodium potassium ATPase, your calcium ATPase, your potassium hydrogen ion ATPase, what have you. Others are secondary active transporters. These will utilize the energy stored in concentration gradients. So because cells pump so much sodium from the cell to the outside of the cell, there's the reason why sodium is the most abundant ion in serum. Cells can actually bring sodium inside cells and piggyback the import of something else on top of that, since sodium is coming down its concentration gradient. So because these molecules are coming in at the same time, they're both going the same direction, that's called a symporter. Or you can send things the opposite direction, like in the case of the chloride bicarbonate antiporter. And this again is using concentration gradients to your advantage. So symporters are used extensively in the digestive system to take up sugars and amino acids and other types of things. Antiporters are used in lots of other systems as well to rid ourselves of things or take up things we want while getting rid of other things we don't want. Symporters, antiporters. When it comes to passive diffusion, gases are taken up via passive diffusion, both polar and nonpolar gases. So oxygen comes in, perfectly fine. Carbon dioxide, which is generated as a metabolic waste, that can leave just fine. Facilitated diffusion uses a carrier. Our cells don't use facilitated diffusion a lot. What usually are going to be taken up by facilitated diffusion are sugars, typically glucose. This is a wholly concentration gradient dependent process. This works in our cells because once this gets in the cell, it gets metabolized. So its concentration inside the cell is very low. So facilitated diffusion works quite well for sugars. Active transport, of course, means you pay the piper in the form of energy, in the form of ATP. Diffusion also works, as we said, for small nonpolar molecules, cholesterol, steroid hormones, things like that. Benzene, naphthalene, other types of things that are not very nice for you. Cells can take up molecules by receptor-mediated endocytosis. This is a highly regulated process. The ligand binds a receptor. That bound receptor assembles a series of proteins that deform the membrane. That membrane deformation culminates in the formation of a coated pit. The pit uncoats. It forms an endosome. The endosome fuses with a lysosome, and this thing then gets degraded. So low density lipoprotein particles, LDL, is taken up this way. So is transferrin. Transferrin is the major iron carrier in our bodies. These are two particular molecules that are taken up by receptor-mediated endocytosis. The receptor typically is recycled back to the surface. This, by the way, is a regulated process. This is one way cells take things up. Cells can take things up non-specifically via pinocytosis. Bulk pinocytosis tends to be unregulated. Fluids are taken up this way, but so can various types of molecules. LDL can be taken up this way. If it is taken up this way, it's unregulated. Cells usually will do that if there's too much LDL around, hence the need to keep your LDL levels in check. Big things are taken up via phagocytosis. There are only certain cells in our bodies that can do this, particularly white blood cells. This is a cytoskeleton-mediated process. You ruffle the membrane, deform the membrane, move the membrane around the thing you want to take up. The membrane that will then fuse and form something called a phagosome. The phagosome fuses with a lysosome to form a phagolysosome. The enzymes in the lysosome digest the thing that the cell took up, and then the small molecules are used essentially to nourish the cell. Anything the cell doesn't need is then released via exocytosis. That's phagocytosis. You see that process in white blood cells. So mitosis, as we said, is cell division. The cell makes a carbon copy of itself. You start out with a cell that's 2N. During interphase, the cell doubles its DNA. It goes to 4N during S phase. The cell then divides, makes a carbon copy of itself, and now you have two cells, both of which are diploid, 2N. And then during interphase, it goes to S phase and it becomes 4N again, and then divides, it becomes 2N again. So during mitosis, the chromosome pairs, are so-called chromatids, duplicate, and then those chromatids will separate. And because each chromosome makes its own chromatid, when those chromatids separate, you still have two copies of each chromosome, hence the diploid 2N. Some cells are capable of taking their resting membrane potential that's generated by the activity of the sodium-potassium ATPase and essentially making themselves more positive. They do this because they have these things called voltage-gated ion channels. Ion channels. So during depolarization, you have opening of the voltage-gated sodium channels. Their inactivation gates will shut somewhere around here or so. And then they will close for good here. And the voltage-gated potassium channels will then open en masse. They started to open here, but they opened kind of with a vengeance here, while the voltage-gated sodium channels close here. You then start to repolarize the cell. It becomes more negative as potassium comes into leaves the cell. And the cell kind of overdoes it. That's called hyperpolarization. But as those channels close, the sodium-potassium ATPase returns the cell back to its resting potential, and then the cell is ready to go again. So this RMP, that means resting membrane potential. The neuron usually needs to get to minus 55 millivolts or so. Here the picture, it says it's minus 65, but it's in the vicinity of 65, minus 55. That's called threshold. It's at here. If you get the axon hillock to the threshold, you have an all or nothing potential in the axon. The dendrites, cell body, they are only capable of graded potentials that are local, but those can add up. And if they add up at the hillock to threshold, you get the all or nothing potential in the axon. I won't belabor this because you went over this in your physiology class, but cells use glycolysis to degrade glucose to pyruvate. Pyruvate gets sent to the mitochondrion, where it's converted to acetate. Acetate is bound to coenzyme A. In the Krebs cycle, that acetyl coenzyme A is completely converted to high energy electrons that are given to these carriers, NADH and FADH2. And this will also produce carbon dioxide and water. These electrons, these high energy electrons, you need to oxidize these carriers to keep oxidizing glucose or fats or whatever. These electrons, therefore, will go to the electron transport chain. When passed through the electron transport chain, you pump protons into the intermembrane space. Those are used to power the ATPase to make ATP and to make a whole lot of it. And that's the process of cellular respiration, otherwise known as oxidative phosphorylation. Fatty acids go straight to the mitochondrion. They're converted via a process called beta oxidation to acetyl coenzyme A. From there, those acetyl coenzyme A's will enter the Krebs cycle, and you repeat. So fatty acids will also go straight to the mitochondrial matrix. So it's free fatty acids, they go straight to the mitochondrial membrane. And they're degraded by beta oxidation. Amino acids will enter either glycolysis or they'll enter the Krebs cycle. Regardless of where you enter, you still make high energy electrons that go to the electron transport chain. All metabolic roads leads to Rome, so to speak. Cells can signal. A cell can signal to itself. If it does that, that's called autocrine signaling. You see autocrine signaling in the immune system. T cells, for example, make interleukin-2. That generates T cell growth. It also activates itself. That's autocrine signaling. More commonly, cells will receive signals from other cells. Small molecules will diffuse through the medium, bind to receptors, and activate recipient cells. This is called paracrine signaling. Paracrine signaling is typically over a short distance. This type of signaling, whereby this is dumped into the bloodstream so that it's carried a long distance and then activates cells far away, that's endocrine signaling. So with paracrine signaling, these signaling molecules are not put into the cell circulation. Endocrine signaling. So epithelial cells are attached to basement membrane. The components of the basement membrane is extracellular matrix. Extracellular matrix consists of lots of different molecules, so collagen, proteoglycans, fibronectin. Fibronectin binds to cells via integrins, other things called laminin. All of these types of molecules play important roles in the lives of the cells, and different types of extracellular matrices can be specific for different organs, and they play different roles in cell regulation. Also, the matrix will change if the tissue is damaged, so the tissue can repair itself. Then when it's time for cell division to stop, the matrix changes. That sends a signal to the cell to stop. These integrins are really, really central in regulating the signals that get passed between the extracellular matrix and the cell. Chromosomes have some parts to them. There's a part, it doesn't have to be near the center, but it's often drawn near the center, called the centromere. The centromere doesn't have any coding DNA, but this is a site where you assemble the kinetochore. And the kinetochore is important for cell division because the kinetochore proteins will attach the microtubules that pull the chromosomes apart during cell division. At the end of the chromosome, you have these things called telomeres. These telomeres are caps on the chromosomes are extremely important for chromosome stability. Aging seems to be involved with fading of the integrity of telomeres. There's an enzyme that helps make the telomeres. It's called telomerase. And telomerase turns out to be a target for specific types of cancers. There are drugs that inhibit telomerase specifically. And the stretch of DNA, as you can see, some of those stretches encode proteins or maybe an RNA, and those stretches are called genes. So genes are found in the DNA, and the DNA composes chromosomes.